Hello everyone, and a very warm welcome to this Science SSS and Science Signaling webinar entitled Part 3, Targeting Cancer Pathways, Tumor Metabolism and Proliferation. Thank you for joining us today. I'm Sean Sanders, Editor for Custom Publishing at Science, and I'll be moderating this live webinar event. As the title denotes, this webinar is the third in a series focusing on the cancer pathways that support tumor development, the emerging research in identifying and targeting these pathways, and innovations in the development of increasingly efficient cancer therapy options. The first two installments on this topic are available at webinar.sciencemag.org. Recent advances in our understanding of cancer have revealed that the disease cannot be understood through simple analysis of genetic mutations within the cancerous cells. Instead, tumors should be considered as complex tissues in which the cancer cells communicate directly and indirectly with the surrounding cellular microenvironment and evolve traits that promote their own survival. In this webinar, we will explore how cancer cells are able to reprogram their metabolic pathways to enable energy production under conditions that are disabling for most normal cells. Of particular interest are how tumor-specific metabolic changes promote oncogenic progression and how these changes can be exploited to develop more efficient treatment options. It gives me great pleasure to introduce our speakers to you now. They are Dr. Celeste Simon from the University of Pennsylvania in Philadelphia and Dr. Nissim Hay from the University of Illinois in Chicago. Thank you both very much for being with us on the line today. Before we get started, I'd like to share some information for our online viewers. At the top right of the screen, you'll find photographs of today's speakers and a view bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information related to today's discussion and the link to download a PDF version of the slides. After the speakers' presentations, we will have a short Q&A session during which they will address some of the questions submitted by our live online viewers. So if you're joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box and clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top right of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology and the Koch Institute for sponsoring today's webinar. Now I'd like to introduce our first speaker, Dr. Celeste Simon. Dr. Simon is the Scientific Director of the Abramson Family Cancer Research Institute of the Perelman School of Medicine at the University of Pennsylvania. Her research is focused on how cells sense and respond to changes in the availability of molecular oxygen and nutrients, with a particular emphasis on understanding the effects of hypoxia in solid tumors. A very warm welcome to you, Dr. Simon.
Thank you very much. It's great to be here. Shall I begin my presentation?
Sure, please go ahead.
Okay. So I'd like to use my time to focus on the unique relationship between tumor metabolism and the reality that malignant cells exist in what's intrinsically a very stressful microenvironment. This includes cells being very limited in terms of access to molecular oxygen. This is a state that's known as hypoxia, but hypoxia is almost always coupled with decreased availability of nutrients, and so we can even think of many domains within solid tumors as more like tumor ischemia. And this is because as tumors eventually achieve a size that extends beyond the natural diffusion limits of either molecular oxygen or blood-borne nutrients such as glucose, glutamine, and lipids, the natural response would be internal domains which cells have, as I said before, deficiencies in oxygen and nutrients, and this impacts many aspects of disease progression, including the acquisition of blood vessels which are needed to sustain tumor growth, and allow tumor cell dissemination during metastasis. Of note, going from the small avascular tumor in the upper left-hand corner to the large, more vascularized tumor in the right-hand corner can actually take several decades based on rates of mutation studies performed in pancreatic cancer. But nevertheless, once the tumor does obtain blood vessels, it can in some way continue to grow and ultimately metastasize. The problem is, as shown on my next slide, that tumor blood vessels differ from their normal counterparts by a number of key features, and they're listed here. First of all, they lack the typical arterial capillary venule hierarchy. They display aberrant, somewhat loose associations with parasites, as shown in the slide. They're highly fenestrated. They can be tortuous, exhibit excessive branching. They can abruptly open and close, and the result is chaotic blood flow patterns and very poor perfusion of the tissue. And this is highlighted in the next slide. What's shown here is a section of a fibrosarcoma taken from a patient that was treated at the University of Pennsylvania and administered the nitramidazole compound called EF5. And there's several important points to make from this slide. First of all, the tumor indeed does have a number of blood vessels, and this is shown by immunostaining for the endothelial cell protein, alternatively known as PCAM or CD31, and that is shown by the green fluorescence depicted in many parts of this fibrosarcoma section. And adjacent to the green blood vessels are cells that are reasonably well oxygenated, but not that far away are cells that are clearly quite hypoxic, and that is because the nitramidazoles introduce thiol adducts on proteins and peptides within hypoxic cells. For the reaction to work, these cells must exist below roughly 1 to 1.3% oxygen or 10 millimeters of mercury, and they are now immunoreactive for an antibody that detects these protein modifications. Again, the important point is that even tumors with blood vessels have dysfunctional blood vessels, and the result is domains where cells are limited for oxygen, glucose, glutamine, etc. Now, the oxygen and nutrient-limited cells will respond in a variety of very sensible ways. They will very rapidly limit anabolic metabolism. They will rapidly inhibit an mTOR pathway, which is a pro-growth pathway. They will engage a long list of stress responses, which include autophagy, the unfolded protein response. There are a number of oxygen-consuming biochemical reactions that will change, and there's a growing family of oxygen 2-oxyglutarate dioxygenases, which regulate DNA methylation, histone methylation, and collagen modification, among other important biological processes whose KMs for molecular oxygen will indicate that many of them will also be influenced by the oxygen gradients typical of most solid tumors. Nevertheless, one of the best characterized oxygen sensing mechanisms is depicted here. This is a large transcriptional response regulated mostly by hypoxia-inducible factors or HIFs. And if you're not familiar with these factors, they're dimers. They consist of a constitutively expressed beta subunit, also known as ARNT, shown as the light yellow ball on the right-hand side part of the slide. But they're also composed of extremely O2 labile alpha subunits. And so in oxygen replete or normoxic conditions, the alpha subunits are extremely unstable because they are hydroxylated on proline residues by a family of HIF-specific prolyl hydroxylase domain enzymes called PHDs. Hydroxylated HIF-alpha is now recognized by a multi-protein complex that includes the tumor suppressor protein von Hippel-Lindau, targeting it for proteasome-mediated degradation. As O2 levels decline, the alpha subunit is now reversibly stabilized, enters the nucleus, dimerizes with ARNT, binds to HREs in the genome, and enhances the expression of literally hundreds of genes that contribute to malignant progression. They're involved in metabolism, tumor angiogenesis, rates of cell proliferation, and inflammation. And among the many things that the HIFs do is interface with metabolic pathways that are active in proliferating cells. And what is shown in the red box region are the domains of metabolic pathways that have been traditionally associated with hypoxia and activated HIF. And that includes excessive or enhanced glucose import, because many of the HIF targets include glucose transporters on the surface of cells. Most of the glycolytic enzymes are actually regulated by hypoxia, or they're HIF targets. Rates of oxygen consumption by the mitochondrion are downstream of the HIFs. Glutamine import and glutaminylysis is enhanced by hypoxing the HIFs to allow TCA cycle anaplerosis to occur, maintaining lipid synthesis under low O2. And so, along with other stress responses, such as autophagy, or the unfolded protein response, the HIF pathway has a role in helping to coordinate oxygen and nutrient availability with the rates of these metabolic pathways to somehow allow cells not only to survive in the stressful tumor microenvironment, where they're limited for oxygen and nutrients, but to somehow continue to proliferate. And this is what I would like to talk about today. Now, HIF-1α and the related isoform HIF-2α are found in numerous tumor diagnostic biopsies and typically correlate with poor prognosis, but one form of cancer is particularly dependent on HIF, and this is the renal cancer known as clear cell renal cell carcinoma. Kidney cancers can be subdivided based on their histopathology, with clear cell renal cell carcinoma being by far and away the most prevalent form. Roughly 75 to 80% of renal cancers are this variety. They get their name because upon H&E staining, they have a so-called clear cell cytosol shown in the right-hand image in this slide. And this is because they're thought to excessively accumulate lipid and glycogen. And in over 90% of clear cell renal cell carcinomas, the VHL protein is disabled in some fashion, which means that we thought they had essentially full-on HIF activity, making the HIF central in the initiation and progression of this disease. Now, beyond the VHL loss, however, they're genetically very heterogeneous, and we and others have engaged in extensive molecular characterization. This includes copy number variation studies, exome sequencing, transcription profiles, and other assays of the genomic and RNA content of these tumors. We recently complemented this with a Penn metabolomic assessment. What you're looking at here is a heat map of roughly 420 metabolites, comparing metabolites in kidney tumors relative to adjacent healthy kidney tissue, and we saw a number of consistent changes. For example, the renal tumors had lower accumulation of urea cycle metabolites. They had increased accumulation of long-chain fatty acids, decreased abundance of lysolipids, but the red box regions show the metabolites which were the most different. So the renal tumors had a significant increase in the abundance of metabolites that fall loosely in the category of glycolysis, gluconeogenesis, and glucose-related sugars. We integrated the metabolomic assessment with a metabolic gene set analysis based on RNA-seq data, and what this is depicting is over 2,700 metabolic genes, i.e. they're mRNAs that encode metabolic genes, subdivided into 72 functional groups based on the KEGG classification. And what's immediately obvious and what's somewhat surprising to us is that many of these functional groups, if anything, are underexpressed in the renal tumors relative to healthy tissue. And the most underexpressed category was one involved in carbohydrate storage. The top hit is the catalytic subunit of glucose-6-phosphatase, phosphoenol pyruvate carboxykinase, or PCK1, and then two isoforms of fructose-1,6-bisphosphatase, or FBP1. Now, all three of these enzymes, as shown in the next slide, oppose glycolysis in central carbon metabolism. It's appreciated that many malignant tumors and cancerous cells engage in enhanced glycolysis to complement oxidative phosphorylation, and the three green enzymes, PCK, FBP, glucose-6-phosphatase, would actually oppose this. They engage in gluconeogenesis and glycogen storage. Kidney is a good example of a gluconeogenic tissue. So it's not surprising that gluconeogenic enzymes would be downregulated in favor of the glycolytic pathway. But FBP is the rate-limiting enzyme, and we've been able to now assess over 1,000 individual patient samples and have shown that this is a nearly universal metabolic adaptation. FBP is universally lost at the RNA and protein level. It's mutated, or I should say deleted, about 40% of the time, and so this is something that the tumors do. They want to oppose FBP1 activity, gluconeogenesis, to promote instead glycolysis and glucose-carbon flux into the pentose phosphate pathway to produce not only nucleotides, but reducing equivalents in the form of NADPH. Now, this is hardly surprising, but what is surprising is that FBP1 is another example of a metabolic enzyme that has both a daytime job, that is, it has its metabolic activity in the cytosol, but much to my surprise, FBP1 can also be found in the nucleus. So in the upper left-hand corner is healthy kidney tissue, where by IF immunofluorescence we can show that FBP1 is in both the cytosol and the nucleus. Probably more convincing is the subcellular fractionation shown in the right-hand side, where it's very clear that FBP1 protein is in the cytosol, but also the clean nuclear fraction. Also shown is the fact that we can introduce wild-type FBP1, this is in the lower right-hand side, into these cells. These are renal cancer cells that don't normally have FBP1. If we introduce FBP1 in, we actually saw that a number of canonical HIF target genes that I mentioned previously, glycolytic enzymes like LDHA, pyruvate dehydrogenase kinase 1, the glucose transporter GLUT1, and the angiogenic factor VEGF are underexpressed. So FBP1 is actually opposing HIF transcriptional activity. If we now tag FBP1 with the potent nuclear export sequence shown over in the left-hand side, so there's no longer FBP1 available in the nucleus, the ability of FBP1 to attenuate HIF activity is significantly lost. So the take-home message is that FBP1 is found in the nucleus and can actually oppose HIF transcriptional activity. In fact, FBP1 can physically associate with both HIF-1α and HIF-2α, as shown here by immunoprecipitation. Not shown here is that not only does FBP1 physically associate with HIF-1α and HIF-2α, it does so at the level of chromatin at the HREs of a number of canonical HIF target genes that fall in the category of metabolic enzymes, inflammatory cytokines, angiogenic factors, and other outputs. So to summarize, what I've shown you today is that hypoxia and responses to the hypoxic nutrient-poor microenvironment impose a number of important metabolic changes. Many of these have to do with metabolic enzymes themselves, but as shown in the left-hand side of this conclusion slide, many metabolic enzymes have surprising roles. So once again, FBP1 in the gluconeogenic tissue such as kidney has a role in the cytosol where it carefully balances the toggle of glucose carbons in central carbon metabolism through glycolysis in the pentose phosphate pathway versus gluconeogenesis. But surprisingly, and there are a growing number of metabolic enzymes that fit this category, FBP1 can also be found in the nucleus. And it's actually in the nucleus for a number of reasons, including to restrain HIF activity. And so we thought for some time that the first event in promoting clear cell renal carcinoma is the universal loss of the von Hippel-Lindau tumor protein shown in the right-hand image, But we now know that the second hit has to be loss of FBP1. Now, because of loss of both VHL and FBP1, we have essentially full-on HIF activity. This revs up glucose uptake, enhances glycolysis, shunt through the pentose phosphate pathway, and many other metabolic targets of HIF that sustain tumor cell survival and tumor growth in the naturally stressful tumor microenvironment. In the future, we will be performing ChIP-seq for FBP1. That might be surprising for a gluconeogenic enzyme, but we want to see how many of the HIF HREs are occupied or co-occupied by FBP1. We've been now able to extend our observations in renal cancer to liver cancer by doing ChIP-seq and also performing proteomics and mass spectrometry. We'd like to identify other nuclear factors that FBP1 may be influencing. This may involve, for example, Wnt beta-catenin signaling. We've been able to show that there's a similar relationship between the related factor FBP2 and muscle-derived tumors such as sarcomas. And then finally, we've now established a conditional allele of FBP1 to explore the relationship of FBP1 and HIF and metabolic zonation in liver and kidney, where certain oxygenated regions engage in gluconeogenesis, but poorly oxygenated regions instead promote glycolysis. And we'd ultimately like to combine VHL loss with FBP1 loss to generate a more faithful replica of clear cell renal cell carcinoma in a genetically engineered mouse model, which is something that has not been a benefit to the field up to this time. So thank you for your attention, and I'd like to now turn it over to my colleague, Nissim Hay.
Great. Thank you so much, Dr. Simon, for that very engaging presentation and a great start to the webinar. We are going to move on to our second speaker right now, Dr. Nissim Hay. Dr. Hay is currently a distinguished university professor in the Department of Biochemistry and Molecular Genetics at the University of Illinois College of Medicine. The main body of his research has been to delineate the mechanisms by which the serine threonine kinase AKT affects tumorigenesis and metabolism, both at the cellular and organismal levels. In addition, he investigates the role of glucose metabolism in cell proliferation and survival of cancer cells, and how it could be exploited for cancer therapy. So a very warm welcome to you, Dr. Hay.
Thank you, Sean, for the opportunity to present our studies today. What I'm going to tell you about is how we could exploit glucose metabolism in cancer cells for cancer therapy. So high glucose rate of metabolic rate of glucose is a feature of cancer cells that distinguish the cancer cells from normal cells, and this could be exploited for detection of cancer cells in vivo, selective detection of cancer cells in vivo. For example, using the fluorodeoxyglucose FDG, glucose analog isotopically labeled in combination with positron emission tomography and CT scan. What you see in this slide is a patient with esophageal cancer before and after treatment using this technology. So, if high glycolytic rate is exploited for selective detection of cancer, why couldn't it be exploited to selectively eradicate cancer? Actually, the FDG PET scan is dependent only on two steps of glucose metabolism: the transportation of FDG by the glucose transporters and the phosphorylation of FDG by Hexokinase. And this is because that unlike glucose, FDG cannot be further utilized in glucose metabolism. In fact, the FDG PET scan is more dependent on Hexokinase activity than on the glucose transporter, because glucose transporters are often bidirectional. So if FDG cannot be phosphorylated, it cannot be trapped inside cells. By catalyzing the first committed step in glycolysis, hexokinases determine the flux of glucose not only into glycolysis but also into other glycolytic pathways. For example, the pentose phosphate pathway, which is required to generate ribonucleotide and NADPH, or the exosamine pathway, which is required for glycosylation of proteins. There are 4 major hexokinases expressed in mammalian cells encoded by 4 separate genes, hexokinase 1 to hexokinase 4. There is also a 5th hexokinase that has not yet been fully characterized yet. The hexokinase 1 to 3 are high affinity hexokinases, whereas hexokinase 4 is a low affinity hexokinase, and it is expressed only in liver and pancreas. Hexokinase 1 and hexokinase 2 share structural and functional similarity, and they have the ability to bind to the outer mitochondrial membrane and utilize preferentially ATP derived from mitochondria to phosphorylate glucose to glucose 6-phosphate, thereby coupling oxidative phosphorylation and glycolysis. The binding of hexokinases to the outer mitochondrial membrane is also important for cell survival. We are interested in hexokinase 2 in particular, and this is because both hexokinase 1 and hexokinase 2 are expressed at relatively high levels in embryonic tissues. However, while hexokinase 2 is not expressed in most adult tissues, hexokinase 1 continues to be expressed. What is shown here at the left side of the slide is a immunoblot showing the expression of hexokinase 1 and hexokinase 2 in different mouse tissues. You can see hexokinase 2 is expressed at relatively high level in the heart and skeletal muscles, and also in fat, which is not shown here. While hexokinase 1 is constitutively expressed in most other mammalian tissues except in the liver, which is expressing only hexokinase 4 and not hexokinase 1 or hexokinase 2. However, when normal cells convert into cancer cells, they start expressing very high level of hexokinase 2 in addition to hexokinase 1. And this is manifested by the FDG PET scan. So I would like to address 3 major questions in my presentation today. First, is hexokinase 2 required for tumor initiation and or tumor maintenance? And I will address that in 4 types of cancer: lung, breast, prostate, and liver. The second question is it feasible to systemically ablate hexokinase 2 and to emulate drug therapy without severe physiological consequences? This is an important question because in mice, the germline deletion of hexokinase 2 is embryonic lethal. And third, why is hexokinase 2 expression induced in cancer cells? To address the first question, we employed a mouse model of lung cancer developed by Tyler Jack's laboratory, in which oncogenic K-ras is expressed upon exposure of the lung to adenovirus expressing recombinase. When we analyzed lung tumors in these mice, we found that hexokinase 2 is highly expressed in the lung tumors, whereas the normal lung does not express hexokinase 2 at all. Hexokinase 1 expression doesn't change in the lung tumors. So what we did, we crossed this mice with mice that have hexokinase 2 flox gene, and we asked whether the deletion of hexokinase 2 will impair the development of cancer induced by oncogenic HRAS. So in this generated mice, both K-ras is activated and Hexokinase 2 is deleted. So in the control group, only K-ras is activated, whereas in the experimental group, K-ras is activated together with the deletion of Hexokinase 2. As you can see in the right panel here, in upper right panel, the deletion of Hexokinase 2 markedly extended tumor-free survival, and the lifespan of the mice was markedly increased. The median lifespan of this mice is 265 days in comparison with 175 days in the control group. We markedly decrease tumor burden in this mice, as you can see in the slide, and average tumor size and proliferation index as measured by BRDO incorporation. So Hexokinase 2 is required for the development of lung cancer in this model. And this is also true in human lung cancer. And what is shown here is a tissue microarray derived from patients with lung cancer, and this is immunostaining with Hexokinase 2 antibodies, and you can see high level of Hexokinase 2 in the lung cancer samples in comparison to normal gland. And this is the staining of Hexokinase 2 is associated with the grade of the tumor, and also high Hexokinase 2 expression is associated with poor prognosis of patients. We have generated lung cancer cell line, human cancer cell line, in which we can conditionally silence Hexokinase 2. We then subjected the cells to a xenograft assay in mice. When the tumors were palpable, we then exposed the mice to doxycycline, which then delete Hexokinase 2, and you can see that we markedly attenuated the tumor growth in vivo when we deleted Hexokinase 2. We also find high level of expression of Hexokinase 2 in mammary gland tumors in mice, in several mouse models of breast cancer, RB2-new and polyoma middle T, and the level of Hexokinase 2 expression is almost equivalent to the level we found in normal fat tissues. We therefore used a mouse model in which RB2-new is expressed in the mammary gland together with 3 recombinase. We cross this mice with Hexokinase 2 flox mice, and we ask what happened in the absence of Hexokinase 2. As you can see here, the deletion of Hexokinase 2 markedly attenuated the tumor formation and extended the tumor-free survival. In fact, it is more than that, because when we isolated tumors from the mice without Hexokinase 2, we found that the tumors were derived from cells that escaped a complete deletion of Hexokinase 2. And this is true also in human breast cancer. This is a tissue microarray from patients with breast cancer. You can see high level of Hexokinase 2 expression in the tumor samples, and this is again correlated with the grade of the tumors. And high level of Hexokinase 2 is associated with poor prognosis. Here we have conditionally deleted Hexokinase 2. This is in the bottom part of the slide. we have deleted Hexokinase 2 in breast cancer cell line, and we autotopically inoculated the cells and to develop tumors. And you can see here that in the red line, the tumor growth was markedly attenuated in absence of Hexokinase 2. We then wanted to address the question whether it is possible to emulate drug therapy, namely to systemically delete Hexokinase 2 whole body and to inhibit lung cancer progression. For this purpose, we used a different mouse model in which oncogenic K-ras is spontaneously expressed in the lung, and the onset of tumors is about 8 weeks after birth. We isolated cells from the tumors in this mice, and we deleted Hexokinase 2 in vitro, and we markedly inhibited the proliferation of the cells. So we crossed this mice with mice that have Hexokinase 2 flux allele together with a recombinase that can be induced by tamoxifen, and that is expressed in all cells in the body. So after 8 weeks, after tumor onset, we deleted whole body Hexokinase 2 by injection of tamoxifen to the mice. And after 20 weeks, we analyzed the lungs of these mice. What we found is that here, even after tumor onset, the systemic deletion of Hexokinase 2 whole body decreased tumor burden, number of tumors, and tumor size. Most of the tumors that were developed in the absence of Hexokinase 2 are much smaller, but a subset of tumors have similar size to tumors developed in control mice. But when we analyze these tumors for Hexokinase 2 expression, we found that they are still expressing Hexokinase 2, namely these tumors were derived from cells that escaped a full deletion of Hexokinase 2. Okay. Now I would like to switch to another type of tumors, and this is quite interesting here, liver cancer. As I told you before, the normal liver cells, normal hepatocytes, do not express either Hexokinase 1 or Hexokinase 2. They express glucokinase. But once they develop cancer, when they are converted to cancer cells, they turn off the expression of glucokinase and start expressing high level of Hexokinase 2. This is also true in human cell line, liver cancer cell line. As you can see here, they are expressing only Hexokinase 2, no glucokinase, and no Hexokinase 1. So in liver cancer, there is isoform switch from the high Km Hexokinase, glucokinase, to the low Km Hexokinase 2. And we analyzed tissue microarray from patients with HCC, hepatocellular carcinoma, and we found high level of Hexokinase 2, which is correlated with the progression of the disease. And this is regardless of the reason why this patient developed HCC, even regardless of hepatitis C, hepatitis B, alcohol and non-alcohol mediated HCC. And when we deleted Hexokinase 2 in the liver of these mice, mice are relatively resistant to hepatocarcinogenesis, and the incidence of tumors in these mice was markedly reduced. When we conditionally silence hexokinase 2 in human HCC cell line, we markedly inhibited the proliferation of the cells and the growth of the cells in vivo, here as shown in the upper part of the slide. And we inhibited also anchorage-independent growth by silencing hexokinase 2. And this is a hallmark of cancer cells, and we could restore anchorage-independent growth by re-expressing a wild-type hexokinase 2, but not a kinase-dead form of hexokinase 2, and not by re-expressing glucokinase. The fourth type of cancer is prostate cancer. Here we employed a mouse model for prostate cancer in which the tumor suppressor P10 is specifically deleted in the prostate of the mice. We crossed these mice with hexokinase 2 flox mice, and we asked what happened to prostate tumor development. As you can see in the Western blot here, hexokinase 2 is highly expressed in the prostate of the P10 null mice, and when we deleted hexokinase 2, you can see a good deletion of hexokinase 2, and the deletion of hexokinase 2 impaired tumor development as manifested by the weight of the prostate and the tumor-free survival, which is markedly increased. And when we analyzed the section from the prostate of the mice, we found that the deletion of hexokinase 2 not only inhibited the proliferation of the cells, but also increased cell death as measured by cleaved caspase 3. And when we conditionally silence hexokinase 2 in human prostate cancer cell line deficient for P10, we increase the sensitivity of these cells to other therapeutic drugs. And I would like to draw your attention to etoposide. These cancer cell lines are relatively resistant to etoposide because they have hyperactive AKT. But once we silence hexokinase 2 in these cells, we resensitize cells to etoposide. So this is a summary of what I've just told you, and I would like to emphasize three major take-home messages. First, hexokinase 2 ablation reverses tumorigenesis of human cancer cells despite hexokinase 1 expression. Second, during hepatocarcinogenesis, there is an enzyme switch from glucokinase to hexokinase 2, and hexokinase 2 expression is correlated with progression of HCC. And most importantly, systemic whole-body deletion of hexokinase 2 in the mouse does not exert an overt phenotype, but inhibits tumor progression in mouse models for lung and breast cancer. And this is a genetic proof of concept that inhibition of hexokinase 2 is well tolerated and therapeutic for lung cancer. And I would like to thank the people in my lab who did the work that was pioneered by Krushna Patra, and the work on prostate cancer was done by Veronique Nogueira, and the work on liver cancer was done by Danielle DeWal, and these are my collaborators. And thank you for your attention.
Wonderful. Thank you very much, Dr. Hay. And thank you to both of our speakers for those excellent excellent presentations. We're going to move right on to questions submitted by our online viewers. Just a quick reminder to those watching live that you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. So the first question that I have, that I'm actually going to put to both of you, and let me start with you, Dr. Simon. Are there specific cancers associated with specific changes in certain metabolic pathways, or do you see the tumorigenic effects on metabolic pathways are more general?
Well, I think that's a really excellent question, and I'm afraid the answer is it depends. So one thing that's very interesting about renal cancer, or at least the flavor that we're looking at, is that even though they're genetically very heterogeneous, in fact, even in a single patient, subdomains of the tumors have extensive genetic and transcriptional heterogeneity, we are finding common metabolic adaptations. In fact, the loss of FBP1 appears to be nearly universal. Another pathway that's almost equally downregulated are components of the urea cycle, and this is something that we're seeing in each of the nearly 1,000 patient samples we've been able to query in this regard. However, work from Ralph DeBerardinis' lab on non-small cell lung carcinoma shows that even in a single tumor, let alone tumor to tumor, their dependence on glucose versus glutamine versus fatty acid beta oxidation can be quite variable. Coming back to some of the things that we're looking at with regard to, for example, urea cycle enzyme changes, a very significant inhibition of gluconeogenesis, which we think are very particular to this tumor. And then another area that we're working on is their excessive accumulation of lipid droplets. The lipid droplet phenotype is sort of an extreme case of this that we think is somewhat unique to renal cancer and forms of ovarian cancer and sarcoma, but not necessarily as pronounced in other kinds of cancers. The effects on gluconeogenesis we think are more particular to tissues that tend to be more proactive in this regard. So I'm afraid, as is very typical the case, some things are nearly universal. I think many cancers are going to be somewhat glucose dependent, but others are going to be more glutamine dependent. It's going to depend on the oncogenic background. It's also going to depend on, in a single tumor, the oxygen and nutrient gradients I tried to emphasize. So I think the honest answer is some things are going to be more universal. Some are going to be more particular to the tissue type and the oncogenic stress. And then even within a single tumor, it's going to depend on local oxygen and nutrient availability.
Dr. Hay, any comments from you on your experience?
Yes. Actually, Hexokinase 2 is highly expressed during hypoxia. It is a target of HIF, and I guess the cell would be dependent on Hexokinase 2 expression under hypoxic condition, in particular because they switch to glycolysis and they don't have much oxidative phosphorylation.
Great. So another question I'm going to put to both of you. Let me stay with you, Dr. Hay. How do you imagine your work translating into potential treatments, and what sort of time scale do you think you're looking at?
Well, I don't know the exact answer to that. This is dependent on pharmaceutical companies who are interested in developing specific drugs. The major challenge is to distinguish between Hexokinase 1 and Hexokinase 2, but I think this is possible.
And, Dr. Simon?
Well, so there is activity, very intense activity in therapeutic interventions in a number of very widely studied metabolic adaptations. A good example is in the kinds of cancer, AML, glioma, chondrosarcoma, I can't remember the name of this tumor. Anyway, let's just stick with AML, glioma, chondrosarcoma, which have mutations in two different forms of isocitrate dehydrogenases. Very potent selective drugs have been developed by Agios and other companies. They're in clinical trials and seeing promising, very impressive responses, even complete responses in a subset of patients. So those kinds of things are happening, as well as c-MYC and n-MYC dependent tumors. Glutaminase inhibitors have been developed. They look very promising in preclinical models, and they're making their way through a number of early phase clinical trials. In the setting of renal cancer, I think it's a very interesting point because, as I mentioned, many of these metabolic pathways, if anything, are downregulated. FBP1 and the urea cycle enzymes being very good examples. So they clearly contribute to the tumor phenotype, but they're gone. And so we're pressed with the conundrum of how do you drug something that isn't there? And there are ways to do it. What you have to find are consequences of the absence of these metabolic enzymes. But now one thing we're engaging in, which I think is a very innovative approach, is there are large drug screens that have been conducted on a number of cell lines with expression profiles available. So can we find a drug that provides an expression profile, mRNA expression profile that mimics FBP1 add back, for example, in renal cancers? You know, can we come up with a drug that mimics FBP1 add back or reintroduction of urea cycle enzymes? And so when, as in the case of Hexokinase 2 or mutations in isocitrate dehydrogenase or over-reliance on glutaminase are available, those things can be drugged. If we're looking at something that's missing, we have to do something a little bit more innovative, I would say. But those means are available now.
Fantastic. So one of the viewers has said that and made the statement that many of the same metabolic features found in cancer cells can also be found in fast-dividing normal cells, and obviously this is a current challenge in chemotherapy. So if metabolic pathways are to be targeted for cancer therapy, is there a way that one could avoid perturbing normal tissue through maybe more specific targeting? Dr. Hay, I'll come to you with that one.
Okay, as you could see from my presentation, when we systemically deleted Hexokinase 2 whole body in the mouse, the mice did not have any adverse phenotype. So this is a proof of concept that an enzyme could be deleted the whole body without adverse physiological consequences, and yet could be therapeutic for cancer. In terms of Hexokinase 2, as I mentioned, it is expressed only in limited number of adult tissues. And because it's a high affinity hexokinase, even if we ablate 90% of its activity, it will be sufficient for normal cells. But this will not be sufficient for cancer cells.
Sean, may I weigh in?
Sure.
Okay, because one thing that I neglected to mention is that there are nodes upstream of these metabolic pathways, and the HIFs represent a good example, and this is very much along the lines of what Nissim was just mentioning. So we have found through extensive genetic studies in our hands, now albeit mostly with mice, is that the HIFs are extremely critical prenatally, but postnatally, unless you develop a severe pathological situation, for example, we can globally delete either HIF-1, HIF-2, or both postnatally in mice, and the mice, again, very reasonably can tolerate this. Now, these are transcription factors which have been traditionally more difficult to target, but through very extensive structural work, at least HIF-2α dimerization with ARNT now has been very selectively targeted. The form of cancer I just discussed, clear cell renal carcinoma, is uniquely dependent on HIF-2α, and so it's going to be, I think, quite successful to inhibit HIF-2α upstream of many of the metabolic pathways that I've just been discussing. I mentioned the lipid droplet accumulation. It interfaces with FBP1 loss and changes in urea cycle and lipid droplet accumulation. And so we actually, and this is being borne out in early phase clinical trials, even though this is a central factor, we believe there is a reasonable therapeutic window. Of course, when you have the unfortunately somewhat more rare case, but a number of patients affected by, for example, isocitrate dehydrogenase mutations, fumarate hydratase mutations, succinate dehydrogenase mutations, now you have the unique opportunity, more or less like what was accomplished with BCR-ABL and Gleevec, where there was an activity that was very specific to the malignant cells not shared with the normal.
So let me stay with you quickly, Dr. Simon, for a more specific question. One viewer says that in their experiments, they see an increase in hypoxia, but not an increase in HIF-1 or HIF-2 protein levels. And they ask whether hypoxia always correlates with a change in expression of the HIFs in your experience.
In my experience, the answer would absolutely be yes. HIF-1α is ubiquitously expressed. HIF-2α is more tissue restricted. By and large, this is not a change in mRNA level. It's a very rapid, reversible change in stability where the half-life goes from less than 3 minutes to 150 minutes. But it depends on the level of O2. In our hands, O2 levels have to decline below, shall we say, 3% O2 to really see a robust response. I'm wondering if perhaps in the experimental conditions outlined, hypoxia hasn't really been achieved.
Right, Dr. Hay, a couple of questions for you. Have you determined the subcellular localization of HK2, and specifically this viewer is interested in lung cancer cells, whether HK2 is associated with the outer mitochondrial membrane?
Yes, it is associated with the outer mitochondrial membrane in most cells. In fact, we found that at least in HCC, if we ablate endogenous Hexokinase 2 and express a mitochondrial deficient mutant that cannot bind to the mitochondria, it could not restore the proliferation of the cell, suggesting that mitochondrial binding is also important.
Great. Another question for you, Dr. Hay. Most of the resistance through AKT is caused by negative feedback to proteins upstream of AKT, according to this viewer. And they ask, what is the relationship between HK2 and AKT, and does HK2 play a role in feedback to HKT? Okay.
So AKT can phosphorylate Hexokinase 2, and it could mediate the interaction with the outer mitochondrial membrane. In fact, when you activate AKT, there is more association of Hexokinase with the mitochondria, and this is in part a mechanism by which AKT could promote cell survival. Perfect. Well, that actually brings us to the end of the webinar because unfortunately we are out of time. So it just remains for me to thank both of our speakers very much for being with us, Dr. Nissim Hay from the University of Illinois and Dr. Celeste Simon from the University of Pennsylvania. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science and Science Signaling available at webinar.sciencemag.org. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We would love to know what you thought of the webinar. Send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel for their wonderful presentations and Q&A discussion, and also to Cell Signaling Technology and the Koch Institute for their kind sponsorship of today's educational seminar. Goodbye.